Hi, as said, my name is Cecilia Unoson and I work as Head of Application Research at Bioworks. And now I will switch topic a bit and I will talk about virus purification. And I will present a study that we have done together with a group at the Helsinki University. But first I want to introduce the Bioworks, even though it was partially introduced recently. So we are a biotech company that is located in Sweden, but we also have sales reps located in other countries, such as, for example, here in Germany. What we do is that we produce and sell agarose-based chromatography resins. And we have a variety of different resins, such as ion exchangers, affinity resins, sec resins, also custom-made resins, so we can target many different molecules. And our resins are called work beads, and they are based on a 45 micron bead. And we believe that this is a sweet spot to do purifications both at lab scale as well as bioprocess scale, because you do get a good separation at a relatively modest back pressure. And those are also the resins that I have used throughout this study. So when it comes to viruses and virus-like particles, they are very complex nanoparticles, and they can vary in a lot of different properties, such as, for example, size, shape, mass, density, but also in the coat structure, so they can have different charge distributions and different hydrophobicities. And those are also the properties that we exploit when we set up different virus purification setups. And to be able to target as many viruses as possible, you need to use different approaches. So we had a group that approached us because they did virus purifications, and they were not happy with the setup they had, because they use like this more classic virus purification. So it consists of a lot of steps, like filtration steps, precipitation steps, and a lot of ultracentrifugation techniques and other centrifugation techniques. So they found this to be time consuming and also very labor intensive, because they were a lot of different steps that could not be connected. They also got too low yields and it was difficult to scale up. So they needed a more efficient approach and preferably a more automated approach. So together with them, we developed like an inline chromatographic setup. So you have like an anion exchange step with a sec step that you connect together, so you can run everything in a single flow. And the virus that you wanted to purify is a bacteriophage called PRD1. And it's a membrane-containing tailless icosahedral double-stranded DNA virus. So you can see an illustration of it here. And it's medium-sized. It has a diameter of approximately 65 nanometer and a mass of approximately 66 megadalton. I should also say this is a well-established virus, because it do share many similarities with adenovirus. So they have the coat structures that are evolutionary related. And as you can see here, they share an overall architecture. So they have identical capsid lattices and attachment proteins at their vertices. So this is the actual overview. So those two setups are the setups that we wanted to compare. So we have setup 1, and this is what they did today. So they had two main steps. You have the first step, that is a PEG precipitation step. And then you have a second step that is a density gradient ultracentrifugation step. But here you also had to add a concentration step after. And then you have our developed chromatographic setup. So you have your anion exchange step that is connected to a sec step, and you can run it inline in a single flow. So this is the actual study setup. So anion exchange it has commonly been used as a chromatography technique when it comes to virus purifications, because you exploit the negatively charged viral surface proteins. But when it comes to sec, you use the property that the virus is macrosized. So it will elute in the void volume, whereas the impurities that are smaller, they will be retained in the resins since they can enter the pores. So we looked at different parameters in this study. So we started to look at the actual sample feed. And in the virus lysate, you have a lot of whole cell chromosomal DNA. And this tends to make the feed viscous. And it's also very challenging for the downstream purifications. So after some initial studies, we determined to use the DNA-treated lysate. And the next thing we thought about was if we should use the DNA-treated lysate and do a PEG precipitation of that lysate. Because PEG precipitation is a very efficient technique, but it's not good enough to have as the only technique. So we thought we might combine PEG precipitation with SEC instead and omit the anion exchange step. But after having done a lot of different experiments, we could conclude that it was not good. Even though we got a high yield, the purity was compromised. So we decided to, throughout the study, use the DNA-treated virus lysate feed. And the next thing we looked at was the actual optimization of the individual chromatographic steps. And then we had our anion exchange step, where we first looked at the binding capacity, so we knew how much we could load. And then we optimized the elution conditions, because we wanted, of course, to have as good separation between the virus and the impurity species. Next, we went to the SEC step, and there we looked for the optimal exclusion limit of the resin to use. So we looked at different pore-sized resins. And we further looked at the sample load, because we needed a certain loadability to be able to have it in our inline setup. And once we had them optimized, we went for our inline setup. And there we looked for reproducibility, yield, purity activity, and productivity. And here, importantly, we also compared them to the other setup, the more traditional setup. And I should also mention that as running buffer, and we also call this the virus buffer, we have 20 millimolar potassium phosphate, 1 millimolar magnesium chloride at pH of 7.2, and we also supplement this with 1 molar sodium chloride in the elution buffer. So now we come to the first thing we looked at for the anion exchange resin. So the resin we used is called Workbees 40Q. It's an agarose-based strong anion exchanger. And we did a DBC measurement, dynamic binding capacity. We did this at a residence time of 4 minutes. And we used the lysate as feed because we did not want to use purified viruses, since we needed all the impurities to be present, since they will also compete for the ligands. And we determined the DBC by frontal analysis at a 10% breakthrough. So here you have the actual experiment. So we first determined the feed activity, and it was determined to be 1.2 times 10 to the 11 plaque-forming units per ml. And this feed was later then loaded onto our resin, and we can see here that everything binds. So we collected the fractions and performed plaque assays. But after a while, you start to see a breakthrough. And the DBC value was determined when you reached a value of 10% of the activity that you had in the feed. So the DBC value under those flow rates were determined to be 23.6 ml of virus lysate per ml of resin. And this corresponds to 2.8 times 10 to the 12 PFU per ml. So now we knew how much we could load onto the resin. And then we went on to optimize actual elution conditions. So what we did was that we loaded 40 ml of virus lysate, and that corresponds to 50% of the resin's DBC. And you can see here, we load it, and then we elute with a long linear gradient. And this is over 30 column volumes, and we do see some separation. And after having conducting plaque assays, we can determine that the last peak was actually the peak that contained the viruses. And we can also see that this eluted at 0.4 molar sodium chloride. And we did not detect any significant amount of virus activity in the earlier peaks. So we had a nice indication now, and based on those results, we set up a step gradient, because this was needed for our next inline setup. And then we do the same thing. We load 40 ml of feed, but here we instead elute with a pre-step of 0.25 molar sodium chloride. And this is then to elute the earlier, or the loosely bound impurities, whereas we elute the virus at 100% of elution buffer. And this was also confirmed. Here we had the virus activity, here we had no significant activity. So now we have the anion exchange step that is ready to be connected into our inline setup. And then we moved on to the SEC. And as I said, we started to look at the exclusion limits, because the virus will always elute if it's bigger than the pores in the void volume. And you want to have a good separation between the void volume and the impurities. So we packed columns with a resin or resins with increasing pore sizes, increasing porosities. And we loaded 1 ml of anion exchange pre-purified virus. And you can see here that the virus comes in the peak and the impurities comes well separated. And here you have the smallest pore sized resin to the left, and you increase in porosity, so you go to the largest pore size resin. And what we can see here that the virus eluted in the void, and it was well separated from the impurities, and all of the resins actually compared really good. And you can also get a very high yield using all resins, but we still decided to move on with the macrosec because it does have bigger pores. So in theory, you would expect more impurities, like more large-sized impurities, to be able to enter the resin. And therefore, you could expect a bit purer eluate. But also, you got a bit higher yield using this resin. So next, we went on to the loadability, because we previously loaded 1 ml of virus lysate. But we needed to load at least 5 ml to be able to have it in our inline setup, because this was what matched the elution volume from the previous step. And I should also say that 5 ml on this scale, it's approximately 20% of the resin's column volume, and this is more like a standard for group separations. And also here, you see you get very similar separations. So you do have a very good loadability even at 20% load. So now we had both steps, both the SEC step and the anion exchange step, that were optimized to be connected. So then we moved on with our inline setup. So what we do is that we first recondition the columns and do equilibration, and then we load 50% of the resins DBC on the anion exchanger. And the flow here is directed to waste. So the viruses will bind to the resin, but all the impurities that does not bind will go to waste. Then we have a pre-elution step. So we will elute the loosely bound impurities, and the flow is still directed to waste. So everything will go to waste. Because so far, the SEC column is in bypass. It's connected to the system, but it's not in the actual flow path. But now we want to elute the viruses from our anion exchanger. So we use the 100% step elution step. The viruses are eluted from the anion exchanger. The flow is redirected to the SEC, is loaded onto the SEC, and the virus will then be eluted in the void volume where it will be collected. And then the flow will again be redirected to waste. So you will direct all the impurities to waste. And And this is what the actual chromatogram looks like. So here you have only the anion exchange column connected. Here you have both connected, both the anion exchange and the sex step. And I should say this dashed line here, it is the concentration, and the dotted line is the actual conductivity. But this is a bit hard to see what it is and what's happening. So I will guide you through this step by step. So in the first step, we do the actual loading after having equilibrated the column. And only the anion exchanger is involved in this flow path, whereas the sec is in bypass. Next, we want to elute the loosely bound impurities. So we elute with a step at 0.25 molar sodium chloride. The flow is still directed to waste. And we come to the final step. Then the sec is involved in the flow path. You use a concentration of 100% dilution buffer, and you collect the void volume here. So here you have your viruses, and as you can see, the virus peak is well separated from the impurity peaks, but it's also well separated from the salts. So you do get this automatic desalting as well. And the virus is back in its original virus buffer. And we always see very reproducible chromatograms. We can run this for different batches. It looks really the same. But of course, it had to be analyzed. So what we did was that we looked for yield, infectivity, and productivity. We based this on 4 biological replicates. So either we purified 40 ml of virus feed with the old method, like the PEG precipitation and the ultracentrifugation. Or we purified 40 ml with our chromatographic setup. And here we can see the STS page. So this purple one is the lysate feed. This green one is the virus that have been purified using our chromatographic setup. The blue one is the virus that first have been purified with PEG precipitation, which is followed by a centrifugation step. But this was too dilute, so you still had to concentrate it. So it's this beige lane. So what you should compare here is the green one to the beige one. And you also have all the structural components listed on the right side. So they actually look very similar. And then we went for the actual analysis. And then we looked for yield as plaque-forming units. We looked for specific infectivity as plaque-forming units per milligram of protein. Also purified particles, milligram per liter lysate. And very importantly, the time consumption. So the 2 top rows are the viruses that have been purified using this old method, whereas the 2 bottom rows are viruses that have been purified with a chromatographic setup. And here highlighted is the actual L weight. So if we look at the yield, we can see with the old method, you only get the yield of 13%. Whereas with the new method, you get over 50%. When we look at the specific infectivity, we have relatively similar values. And for purified particles, you have 3.5 with the old method and almost 13 with the new method. And importantly is the time consumption. So using this old method with so many steps that are not connected, it took more than 24 hours. Whereas using this more inline, where you can run everything in the single flow, it could be conducted in only 6 hours. So when we compare the 2 setups, the anion exchange sec setup gives a much higher yield without compromising the infectivity, and you also get a much higher productivity. And also, we do get this automated setup that this virus group wanted. So for conclusions, what we have done is to develop an inline chromatographic setup consisting of an anion exchange step that is connected to a sec step. And And we do see a 3.5 times higher yield increase, and without then compromising the activity, specific activity. And we have reduced this multi-step process into a rapid single flow process that can be conducted in only 6 hours. And it's more than 4 times faster. It's easily scalable, and you can choose if you want to connect it inline, as I presented here, or if you want to run it as individual steps. And we believe that this method is highly promising for similar viruses, for example, adenovirus. And if you want to read more about this, it is published in Journal of Chromatography B. And with that, I want to make some acknowledgements. I first want to acknowledge the Bioworks Applications Research Team, which I call the Chromatography team. And there specifically Mikael Andersson Schön, he did the majority of the experiments. I also want to thank some other people at Bioworks. Lars Haneskog initiated the study, and Anna Hebel and Jan Berglöf, they helped me with proofreading of the manuscript. And then I also want to thank the Helsinki team, which I call the virus team, and then specifically Hanna Oksanen, who did the majority of, or she was the major drive behind this project. She coordinated most things. And then, of course, I want to thank you all for listening, and I'm open for questions. Thank you.